Let's Hi, Tamir.
Hmm.
Vivian. All right, you all exist. Good. That's great. I always get sort of nervous because you know what it's like, right? I'm talking to names. All right, I'm going to give you all a crazy class um, because I like to do this sometimes. And it's based on something Ho Jones said earlier that a lot of biologists, if it's not their job to know genetics, they don't. And so I'm just going to talk about genetics and genomics and things I think are fun and a bit about insects and we'll sort of see how it goes. Um, this might make a lot of sense. Like say you're an ecologist, you maybe haven't taken genetics since high school, you didn't need it for your degree, it was a waste of time. Maybe you do olfaction and you're a neurobiologist, if you're not working in Drosophila or a genetic model, you may not ever find out a lot about genetics or genomics. Maybe you're in social determinants of healthcare and you're trying to work out how access to doctors affects people's health. Genetics isn't going to come into it. So this talk's just sort of talking about things I think is fun in genetics, right? And oh, hopefully this is the right place to start. So can you all see this?
Yes.
All right, good. All right, someone has a voice too. Um, all right. So this is what, why don't people really care? So genetics, genomics, hereditary and variation of inherited characteristics. Like I said, a lot of people fail, I feel, to understand the scale of its importance. This year, we've had 20 years since the human genome was released. 30 years ago, nobody knew any of the things I'm talking about. Just none of them, right? They suspected, but they couldn't do it. The technology wasn't there. Everything I'm talking about today is basically, I'm not going to say new, but our lifetimes. So everything is 10 to 20 years old. Some things are only a few years old, right? I think there's a lot of reasons for people not wanting to care about genetics. Firstly, a lot of biologists, right? They go into the field to go collecting, right? And their idea of fun was not biochemistry. A lot of biochemists find molecular biology kind of easy, right? But if you wanted to be a biologist, maybe you wanted to go to a zoo. Secondly, there's kind of a lot of compute involved, and that's because genomes are big. It's just hard to get your head around all of these genes. I grew up really as a Drosophila researcher, which gave me a huge advantage when it came to genetics because Drosophila is the key, I think probably the most important genetic organism in terms of connecting genotype to phenotype in a multicellular organism. And then finally, changes in genotype are read or acted on by cells in most parts of the body. So sometimes you'll get a change in a gene. It'll be very hard to understand what it does because, I don't know, maybe it makes the arteries in the heart a little bit wider or a little bit skinnier. But from the outside of the mouse, unless you know to look there, you will never find out, right? So often it can be quite hard to understand what's going on. So there's a lot of confusion of like, what the hell does this genetics thing even mean? There's a bunch of changes. You can't even tell me what they mean. But I think that world's going to slowly change and has been changing. So what do I want to tell you? Okay, and I'm going to have to move some photos around. The main things I want to tell you are like, one, I think genomics and genetics, and especially gene content, is the unifying data of life on Earth, right? So we can literally read the history of life on Earth. I'm also going to tell you that for most people, human population genetics has most of the day-to-day effects on your lives, right? Be it Mendelian disease or sickle cell. I'm going to give you some examples of that. I'm going to tell you the last 20 years of technology has made all of this pretty easy and cheap. I'm going to think about why. It's still hard to predict traits from sequence. If you gave me Tom Cruise's DNA sequence, I couldn't tell you what he was going to look like. If he had an identical twin, they would both look the same from the same DNA sequence, right? We haven't got that good yet, but getting better, and I think with AI in the future that might happen. And then finally, the concepts are trivial, right? So let me go a bit more. Ah. So I'm going to start with the concepts. Everything you need to know is basically the central dogma. There's loads of other stuff, but this is it, right? It's just the central dogma is all you need to know. You learned it in high school. And here's my little sketch of this central dogma. Right, we got cell replication. The DNA gets replicated. From there, you get transcription. So you get long pieces of message RNA and heteronuclear RNA. The heteronuclear RNA gets processed. Introns get taken out. You get a poly A on the end. You get a cap. This is assuming you're a eukaryote, right? That message RNA then gets moved out of the nucleus. It goes into translation in the cytoplasm. You have a ribosome. Transfer RNAs come along and they align in the ribosome, they align up along the message RNA and then they link amino acids together and you get a protein. Right? This central dogma is basically all you really need to know about genetics, right? If you've got this, you've got 80, 90% of that. There's a whole bunch of other things. There's a lot of genes, there's a lot of chromosomes, there's a little bit of a genome inside the mitochondria. You know, you get X-linked diseases because of XY. So X-linked diseases are found in males more often. But fundamentally, all you need to know about genetics is this. And then if you need to isolate DNA, you can buy a kit from QIAGEN. If you need to sequence DNA or RNA, you can just send it off to a lab or buy some sequencing stuff. The techniques have become relatively easy. If you want to amplify a bit, you can design primers and do a PCR. And Herman will talk about CRISPR later. The numbers, however, make it a bit more difficult, right? So we've got our 6 locus genomes. Each of these 6 species have 9 gigabases. That's pretty big. The human is 3 gigabases, right? These species have 13 chromosomes. The humans have 23 chromosomes, hence the name of the company 23andMe. There's 80,000 genes, but only 17,000 of them are coding, right? That make proteins. In humans, it's about 23,000 genes make coding proteins. In mammals, especially, yeah. Here you've got the codon table. So, you know, AUG is methionine is a start. You've got stop codons, UAA, UAG, UGA. Of course, U's are in RNA and T's are in DNA, right? And if you think about it, it's quite interesting, right? If you change a gene expression, maybe you knock it down, you can wait a day and you can see a difference in the protein level. And it turns out that most proteins are turned over about once a day or so. And so there's a lot of energy by going into remaking messenger RNA and remaking proteins. And that's why if you knock down a mRNA level, the protein level will drop maybe 24 hours later or something, right? Depending on which gene and which species. But turnover of proteins is really quite fast. And so this happens a lot, this cycle. That's a lot of data. Right? 80,000 anything is 80,000 pages in a book would drive you crazy. Right? So you have to use computers. All right. That's trivial here. Our NCBI GenBank submissions for the locus BPRI genomes. Here's one for Americana. We've got amazing genomes due to this new long read technology, but fundamentally we're getting pieces of sequence, we're sticking them together as a jigsaw puzzle. The tricky part is in the analysis. All right, I think going to switch over. So now you know everything you need to know about genetics. There are details, there's Mendelian stuff, there's quantitative traits, but fundamentally, that's the thing you need to know. And you were all taught that in high school. Never be afraid. It does get, there's more things to learn, but it doesn't get a lot harder than that. Right. So if I was a student, I just never be afraid of genetics. You know, maybe you're going to have to learn which disease is caused by which gene, but that's just sort of a detail you have to learn. I think the other way we have to think about life is, since the human genome, what's affected us. So there's different scales, right? We can have sickle cell, which is a Mendelian trait, right? So there's been a single base pair change in beta hemoglobin, and that means that the red blood cell containing that hemoglobin becomes a bit stiffer and can't fit through holes as well. And this goes through families. And of course, the story is that heterozygous is protective against malaria because it also gets harder to suck up into the mosquito. And homozygous mutations are not so healthy. We can also look at height of humans at birth. This is from the All of Us data. And people have done GWAS where they looked at all the differences. And for every difference in the human genome, they simply asked, do people with that difference, are they particularly taller or shorter than people without it? And they control for sex and they control for ancestry. It used to be that these kind of things didn't work very well because they had low power, but this particular paper saturated the human genome by looking at 5 million people. They found about 7 or 8,000 SNPs that affected height, and they could predict a fair amount of height. That's fairly new. And then on the right-hand side, we're talking about chromosomal changes. This one is trisomy 13, so this leads to Down syndrome. If you look at human births, the main cause of miscarriage, or one of the major causes of miscarriage, is chromosomal aberrations. Okay, so let's try and ask some questions. So here's a question you can ask. What sex will my child be? You're pregnant. Maybe you know. Am I related to Genghis Khan? Probably, maybe not, maybe. But we can now answer that question today. Do I have an inherited Mendelian disease like cystic fibrosis, or like we just said with sickle cell? What kind of cancer is that tumor? What kind of microbes are in my gut, right? We can sequence all the bacteria in your gut and identify them. And then people can start companies saying, oh, that's a good bacteria, that's a bad one, take my yogurt pill, please. I don't know how much that helps people, but it's a trendy thing to do these days. What kind of virus is in the sewage plant wastewater? That's something we've all learned from COVID. Who left these cells at the crime? Not only can we see if you're related to Genghis Khan, we can identify you, and perhaps we can use ancestry.com to try and get your family name from your genome sequence, and then perhaps plus a location, we can actually identify who you are. How many kinds of animals live in the watershed? You know, we can sequence the DNA in water. Now, I'm sort of talking about these theoretically, right? Let's look at a few. So this one was one that happened at Baylor College of Medicine in the Genome Center when I was there about 2011, 2012. These two, at the time they were younger children, had very poor muscle ability, right? They couldn't move much. They were always pushed around in a wheelchair. Their parents never knew what was going on. They went to tons and tons of doctors. And then, and there's two parts of this story you got to know. One is they had a really good coach. So they had this coach, and the coach is like, well, they look kind of weird. They look like some people I know who had dopamine deficiency. Why don't you try giving them some dopamine? And you can buy dopamine as a tablet, and it kind of got them 80% better. The other person in their lives was their dad, who actually worked at Life Technology. And so he knew the sequencing people, and he ended up getting them exome sequenced because, you know, they're twins and the two of them had the same thing. And it turned out that they had a mutation in a coenzyme required for serotonin and dopamine synthesis. So as well as supplementing with dopamine, we also supplemented with serotonin, and within, a few months, they're on the basketball court. The story I heard is like 3 months later, he'd actually broken his arm from playing basketball, and that was seen as a huge success, right? And then, I was looking it up last night, and they actually gave a TED talk for a youth TED thing. So this is a very rare example of us finding the knowledge, which is quite easy, right? We can sequence people and usually identify at least 50% of Mendelian disease at places like Baylor College of Medicine and the genetics department. But also the rare bit is having such an easy fix, right? They couldn't make this neurotransmitter. All we had to do is supplement them. Easy. I don't know if you know David Middleman. He's in Houston. He started a company called Othram, and he used to be in the gerontology kind of like ancestry kind of thing. But currently, what he did is he realized that lots of police departments had tons and tons of rape kits and other DNA tests just waiting around because they couldn't afford to do them all, because they were doing them by hand and they were using the old technology. So he started a company to put it on the new Illumina machines with high throughput at low cost. And so then he also, as well as working with police departments, they have a public site called DNA Solves. So if you have a case and you have a DNA sample, you can send them, I think it's like $7,000 and the DNA sample, and maybe your DNA samples, if it's like you want to say this is related to me or related to whoever, they'll do the DNA sequencing and they'll literally try and solve old cold cases, and they'd be doing quite well doing this. So this is now a routine thing. I was going to try and find a slide about non-invasive parental testing. So let's say you're pregnant and you want to know what sex your child is going to be, and maybe you're 12 months pregnant, and you definitely want to keep the baby, or maybe not. Well, it used to be we would actually take cells off the embryo and do a PCR and work out whether or not there was a Y chromosome in the sex. A while ago, someone realized, and by a while ago, I want to say 10 years ago, so this is pretty recent, someone realized that some of the baby's DNA was also going around the blood of the mother, and you could simply take a blood sample from the mother and screen, and then you can do a non-invasive test. And this is done, this is like $1 billion business. This is a routine thing offered now to all women if you want to know the sex of your baby, or if you want to screen for Down syndrome or something like that. This particular slide, though, is kind of crazy. So I read this blog called Less Wrong, and someone, I think Gene Smith is an obvious anonymous pseudonym, but he went through in this article the math to have a polygenically screened child. So what you could do is, if you saved embryos, and maybe you and your partner fertilize, say, 8 or 10 of them, you could sequence them and pick the best of that 8 or 10. And I don't recommend this because it's actually a real pain in the ass. And he goes through the difficulty, like which clinics have the best success rate of collecting eggs, how young you have to be. Then you've got to freeze the embryos. It's better to freeze embryos than it is to freeze eggs. So this is a big pain in the ass, but it gives you an idea of what's possible. For maybe you can genetically choose between 8 or 10 embryos, which one you would like to have for your baby. And of course, in vitro fertilization is a big pain in the ass. And so I mostly recommend, the old fashioned way of just having sex with people you love and then having babies that way. But for perhaps if people have to do in vitro fertilization anyway, and they're young enough and they know who they want to have their children with, you could actually, in theory, select some children, and based on polygenic risk scores, you might be able to increase their health or decrease their risk of disease by, say, I don't know, a third of a standard deviation, maybe 10 or 20%. Not a lot, but you know, there could be quite a lot, right? So this article was very interesting to me, and it sort of went into the practical side of the math, right? Um, I don't know anybody who's done this yet. I do know that at Baylor, for example, this kind of thing for in vitro fertilization has long been offered to avoid obvious chromosomal abnormalities. So if you've got a history in the family and there's a 50-50 chance if you having a particular disease and you've had one child with the disease and you don't want your second child to have this, you know, bad disease, then you can do the in vitro fertilization and you can pick an embryo without the disease, depending on the Mendelian genetics. That sort of thing does happen. And I also know that there is, when if you do in vitro fertilization, people do take multiple embryos and then they look at them and they check them for chromosomal abnormalities to make sure that the ones they implant don't cause a miscarriage. And that's really helped increase the success of IPT over the years. So this kind of thing is now possible, although not really recommended unless you have a disease in the family, in my opinion. But it's interesting to read this article and look through and it's, you know, it's not written technically. Tanya looks like she's going to say something. You muted, sorry.
I'm right that getting the note of that article, I would like to read that.
It's a silly article, and it's kind of an interesting. Les Wrong's well known for AI alarmism, so AI doesn't kill us all. But that particular article I thought was pretty funny. 2019, I don't know if you guys remember, but in China, someone edited the human genome using CRISPR to disable CCR5, which is an HIV binding target in the human genome. Herman's going to tell us about how our locus CRISPR is going and next in the crash course, I believe. What was interesting about this is because it was not done in the correct way, the entire scientific community jumped on them, but it is just a matter of time before human gene editing becomes a regular thing. And again, when you think of what he did for CCR5, they basically add a stop codon. This is a mutation that was already available in the population, so they had a good idea of what it would do, and therefore that protein didn't get made. So I don't know when CRISPR in humans is going to become routine. I don't think it ever really will. I think humans try to move too quickly, and we'll probably find other ways to give us longer lives and be healthier than CRISPR, because, you know, you want something you can just sort of do, right? I mean, if you want if you want your children to be beautiful, you could try a bunch of CRISPR, or you could try plastic surgery, right? One of them's way cheaper and faster and easier, is going to work. Okay. I want to give a quick note about advances in technology while I'm waffling on, and the top left is this classic graph. So you need to know that we started off at around 2001. This was 100 million for a genome based on Sanger reads. This is a log scale, and today we're running around $800. $800 is short read at scale, assuming you've got this NovaSeq 6000 or other Illumina big machine that costs $1 million. It's that cheap because they put a lot of samples on. There's also new machines, and in these last few years, we've had this massive improvement in high quality long reads, which is what gave us a very easy jigsaw puzzle and really high quality locus genome assembly. So when we did that assembly, we were assembling reads that were 20, 30 kb long with an error rate of 1 in 1,000. So 99.9% accurate. So it meant that the genome assembly worked really well. On the bottom left here, I'm hoping you can all see my little pointer. Is that true? Okay. This is basically a hand-sized sequencer from Oxford Nanopore. This little thing in the bottom right here, this is the plug to put in the internet or whatever. So it's really quite small. I think this company is going to bring low-cost sequencing to the public. It's still cheaper per gigabase to use the big machine, but you might not be able to afford it, and you may not have the throughput, right? Who wants a bus? You might just want a sports car. I've thought about this a lot, and I can tell you how sequencing got so cheap. And the answer is the same way that computing got so cheap. Everything got minimized, right? This $100 million every genome, every sequencing reaction was getting done in a microcentrifuge tube, a 1.5 mil microcentrifuge tube. And then we moved to 96 well plates. Sorry, we moved to 96 well plates, right? This started this jump around here was the 454 technology, where we went to doing 1 million reactions at a time on little beads. And then this continued with the aluminum machine, which made the reactions even smaller. So you needed a microscope to see them, and they put them on a microscope slide and had little patches of DNA. And then finally, this machine in the bottom left is a single molecule sequencer going through a hole and measuring the voltage change as the DNA goes through a hole. This miniaturization meant you could do a lot more sequencing in the same reaction all at the same time, right? You can do this with sequencing because the output is A, C's, G's, and T's, right? It's just information. And so therefore you can make a machine that reads these things and generates it, and its output is A, C's, G's, and T's. The output is not a car or a bicycle or anything. And I think this says something about science, right? In hypothesis generated science, our output could in theory be binary. Maybe it'd be a 0.9 or a 0.1, but it could be just a 1 or a 0. And I think this means that science can be automated. I think that's the big lesson that people should kind of think about. As science can be automated, and if anything can be done multiple times, you can repeat it multiple times, and you can make the experiment as small as possible, because all you need to get out of it is a 1 or a 0. Was my hypothesis true or false? Right? It's not like a car where it has to seat 4 people. You can't miniaturize it. I mean, we've gone from a 20 microliter reaction down to single molecule, right? Anyway, so that's one thing I want to say is it's worth thinking about that. We're in the information generating world, and so we can automate and parallelize things a lot. The other thing too is computers weren't very good when we started, and the human genome is about 1,000 page phone books. And, this is just a random Americana gene, but it's quite long. There's a lot of stuff going on. You need computers. And so the computer revolution at the same time has allowed us to look at the data. One thing I think people forget about is most gene models are simply computer generated by aligning RNA-seq data. They're generally very, very good, but often no one's even looked at them, especially for, obviously humans, they have quite a lot, but everything we're looking at is a simulation or a model or virtual. And so you do, if you're going to do some work, you have to check these things. Okay, I have opinions about unifying biology with genomes. So I'm going to switch gear one more time, and I'm going way too slowly. The Rosetta Stone for arthropod molecular biology is Drosophila. This is my car factory analogy I was told during my PhD by Mike Stern, who's still at Rice. And it goes like this. Let's say you're sitting at a hill outside a car factory and you watch the workers go in and out of the car factory every day, right? Maybe it'd be fun if you grabbed one of those workers on the way to work and you tied them up and didn't let them go in one day. And then in this analogy, all of a sudden the cars come out and they're all missing the windscreens. And you go, aha, the guy I tied up was the guy who put the windscreens on the car. And then the next day you tie up someone else. And all of a sudden the cars come out and they look kind of the same, but they don't make any honking sounds when you press the horn. So you go, oh, this is the guy who did the horn. That's basically what Drosophila genetics has done for every gene in the Drosophila genome. Every single one of them is now being mutated, and then we look for the change in the organism for the phenotype. And for a lot of them, we can say what this gene does. So we know what the opsins do. Drosophila genes are named after the phenotype. So one of my favorite stories is a long time ago in the 90s, there was a screen for sterility, male sterile genes, and they did an EMS mutation to knock out a bunch of genes, and they looked at all the ones that were male sterile. And there were about 200 of these, and the female graduate student had to try and name them all based on different names for male sterility. So they had pointless and blanks and gelded and so on and so on. My favorite, which has come up because of recent movies, was one called Ken and Barbie, because those flies had no external genitalia. But anyway, what this means is if you find a gene that's orthologous or has the same protein sequence as a gene in Drosophila, you can look in Drosophila and say, well, what happened when they knocked it out? And maybe that will help you tell you what it does. A lot of genes, if you knock them out, we're not quite sure what happens, because maybe they're involved in robustness to changes of environments, and we don't have that environment in our assays. But I think as many as 2/3 of genes in Drosophila we have a phenotype for, still a third we don't. I'm going to try and put this in the tree of life. So we got 3 domains of life, right? We've got prokaryotes, eukaryotes, and archaea. We got 64 phyla, 146 classes, 1,000 orders, 950 families. This is all described, 180,000 described genera, about 1.5 million described species. There's probably, I would say, 8 million species on the planet, but depending on how you count, estimates go up to 15 million. And different things are conserved. So here I've got the estimated on the left-hand side, the estimated number of ancestral nodes, right? RNA life, maybe it got as far as 100 genes. Luca, the last universal common ancestor to all life, maybe 500, 1,000 genes. The last eukaryotic common ancestor, Lika, maybe had 3,000 genes. The Herbalitarian, so this is animals now, and all of the animals that have a bilateral symmetry, maybe 6,500. The ancestral mammal, maybe 20,000. And then here I got numbers for human, this is the number of protein coding genes for human, Drosophila, and Arabidopsis, the model plant. And as you go back in time, at some point you go back, all you're looking at is, oh, these have similar protein sequences, they must share this protein sequence genes. If you go back to orders, you can also look at things like gene content and sequence evolution in terms of the order of genes on chromosomes between things like beetles and flies. Cis regulatory conservation doesn't come back until you're actually quite closely conserved. So cis regulatory regulation between humans and other primates is conserved, but if you go as far back as mice, you start losing it very quickly. And mice and humans are about, I want to say, 90 million years diverged. So, this is going back in time a lot. And then right at the population level, we have medical genetics, like we've just been speaking about. Just to give you an idea of what this means, here's the same thing again, but on the right-hand side, there is a vertebrate and a fruit fly example, right? So eukaryotes and prokaryotes in the 3 domains, that's bacteria and a tiger and a plant, animals and plants. at the sort of the phyla level. At phyla, we got chordates and arthropods, right? Class level, right? Invertebrates classes are things like mammals and fish and birds and amphibians and reptiles. At the order level, you've got things like carnivores and primates. Orders in insects would be fruit flies and mantises, things like that. At the family level, cats and dogs, their own taxonomic families. That would be Drosophila, and Muscidae is like houseflies and things. And then in general, you different kinds of cats, and in species, you've got tiger and lion or different kinds of fruit fly. So just to give people like an idea, like 300 million years ago, that's the difference between birds and mammals, or insects and centipedes. I've done a lot of genomes over my time, so I'm going to talk about some of the things we found. Here are some of the photos from one of the last big projects I did, where we looked at 30 different species of insects, right? We did not do any locusts or grasshoppers because the genomes were too big. And Hojan asked me, and I said I couldn't afford it. So we did all the small genome things. Same goes for a mantis. And we also did a lot of pests and things. So this is a bed bug, a cockroach. This is the Colorado potato beetle. This one here is actually quite beautiful. This is the glassy wing sharpshooter, which does Pierce's disease in grapes. It spreads a virus. It's a phloem eater. Here's some dragonflies and so on, and some cool spiders and things like this. Now she's in Reno. Tanya might want to look out for black widows in the garage, because I've got them in my garage downstairs. So we decided to look at, with a bunch of collaborators, and I especially wanted thank Rob Waterhouse, who's sort of brilliant, and Greg's gone on to do great things, and Matt Hahn. How does the gene content change across the arthropods, right? So we added 30, we got to 76 arthropod species. Right now, there are far more high quality genomes out there to do this in a much better way. But this is what we had at the time, which wasn't that long ago. And these were all of the species we did, plus some outgroups. And we made a website that you could go to, so you could ask what were the gene content changes, right? So what are the things I want to tell you? So firstly, we did a tree, and we did a tree with genes. It's very hard to do a tree with every gene. If you put in too much data, things get confusing. But what trees tell you is it allows you to estimate when species diverged and how long ago that was, especially if you link it with the fossil record. So that gives you information about what came from what, right? The fruit flies and butterflies, or the diptera, the flies with two wings, came after beetles and hymenoptera. That's something we know. So that's important. When we looked at all of the genes, we could ask which gene families, and I should tell you, right, the way we sequenced the genomes, we did the RNA-seq, we aligned the RNA-seq to the genomes and used various annotation programs to come up with annotation models. Always say models unless you've done it yourself. And then we simply blasted them together reciprocally, and so it's the best blast hit, right? So if protein from species A blast, the top hit is species B, and then if species B is, if you take that top hit and it's the top hit in species A, then often that's how you say it's a gene family and they're orthologous. Now with higher quality genomes today, you might also throw in location on the chromosome as well, if that's possible, and have a better idea, because you'll have no holes in your genomes and things like that. One thing we found is all of the butterflies had the most new gene families. And I think what this means is we need a new model organism in butterflies, because we cannot like I say, we can't knock out those genes with the mutation in fruit flies because they're just not there, right? A lot of these are, we don't know what they do, but some of them were olfactory, some of them were response to stress, and they're order specific. The beetles also had order specific gene families. If I looked at the gene families changing most on the arthropod tree, most of them were TEs, transposable elements that jump around everywhere. A lot of them were in detoxification, digestion. Some were in cuticle metabolism, and a lot of zinc finger families. And problems of doing science at this scale is I can't go in and CRISPR every one of these and work out what it means, but maybe we'll be able to do that in the future. It's also interesting to think about the last insect common ancestor. So here, Greg put together all of the, here's a gene family and how many copies has it got in our representatives of each of these orders. So maybe it had 2 copies in the scorpions, 1 copy in the spiders, 2 copies in the ticks, 4 copies in this amphipod, which is a crustacean. And then in most of the insects, it had either 0 or 2 copies or 1 copy going on out here in the holometabula. Okay. If you look here, this is the last insect common ancestor. So before this, we had no insects, but you can see that this gene was in the common ancestor and before, right? And so he found 9,600 gene families that were present in insects, but also present before the insects evolved. It's pretty easy. I'm hoping everyone's with me there. He tried to correct that for divergence time and gene loss. So he estimates there are about 14,000 genes in the last insect common ancestor. And of course, we can list 9,000 of them, right? We can also do the opposite. Here we got an example where there are gene families and genes, orthologous genes within all of the insects. And here we picked winged insects and not the Diplura, and none of the species before that. So we found 147 new insect gene families that were born during the transition to winged insects. And we looked at them, and some of them, there's wing morphogenesis families, right? That's good. That's flight, larval behavior, development. That's good. Odorant binding, visual learning. Insects got way better at smelling and looking as they started to fly around a lot more. Cutin was needed, so you could have wings. But fundamentally, I don't think we've really gone fully into that list and followed up. We can do other ancestral gene counts. So to the chelicerates, we've got inferred numbers, which we can actually say we can see these genes for sure. And then our estimated numbers. It's interesting, as you get to Diptera and Drosophila, the gene number kind of drops a bit as you get further on, and the ancestral genes, like the more ancestral insects like the BPRI species and Cichocirca, have a higher gene number and a bigger genome. You can also do this. Elias here did it for his PhD on protein domains. One thing I do need to tell you is over evolution, it's much easier to lose things than to gain things. So single domain gain, we only saw 394 of those using Elias's methods. Domain loss, 9,000. So it's easy to lose things that go back as far as the bilateral. And then here, I'm just going to give you some sort of numbers on the speeds. So you can make this tree And the color of the tree shows either domain changes per year or gene gains and losses per year or substitutions per year. Here in the gene gains and losses, mostly it's pretty stable, but a lot of these ants and the bees here, bumblebees, seem to have a lot more. And then over here in substitutions per year, the same, it's a bit more mixed up, and you can see different branches and things and where it's happening. So we can put this on a tree. And again, this is the same thing. These ants here had a massive amount of gene gain and loss. We looked at rates. No real correlation between gene gain and loss and SNP substitution rates and little changes. So there are different mechanisms going on there. I'm going to skip that. I looked for notable gene gain. Obviously, spiders. We have species with silk and non-spiders without silk. So we've got all the silk proteins, toxins, zinc fingers, the lepses, silks as well. But there's lots of stuff that still could be looked after there. And then lastly, lateral gene transfer is everywhere. These are lists from previous projects, but my favorite is, of course, PAFID, because that's got really cool lateral gene transfer. And then the Asian longhorn beetle, going from bacteria in the gut, basically enabled these beetles to eat cellulose. They lay their eggs underneath the bark. Methylation is everywhere. We are going to be looking at methylation in locust. And I'm going to just shoot through this. So as a quick summary of this, is sort of like we found a lot of emergent gene families in the Lepidoptera. We found a lot of methylation, especially in spiders and larger, more ancestral genomes. High rates of gene gain and loss and domain rearrangements in leafcutter ants. Cockroaches turn out to have the most rapid gene family expansions. And I don't know what the numbers are for the locust, but locust is somewhat related, not really, but closer to them than other things here. The last holometabolous common ancestor had 10 emergent gene families, but mostly when the insects went holometabolous, it was easier to find things that were lost than that were gained. And the last common insect ancestor had 147 emergent gene families, some with wing morphogenesis and other things. There's also other things I like. So when we did the annotation for the BPRI, we had our little gene BPRI. These were some of the questions that I encouraged people to think about, to try and connect obvious phenotype to obvious genotype. These are some of the kind of questions that you can try and think about. But there are also questions you can do, like population size. You can estimate that based on population sequencing a little bit. Gene flow, historical population sizes is something you can estimate going back a couple of million years, maybe as far as a bit more. Gene gain and loss and evolution, symbionts, lateral gene transfer. These are all things that you can do today because you can get the sequence information and you can copy other people's methods. We also need genomes for these technical kind of things. But the thing, how big is my colony? Can I predict a structure with AlphaFold? Obviously, the BPRI has done this, right? Ho-Jun's done a lot of work looking at RNA-seq between swarming and non-swarming grasshoppers and locusts. But all of this now, the sequencing is now accessible and cheap and not $100 million like it was 20 years ago. Lastly, I'm going to try and shoot through this because I'm taking far too long. This is the Earth Genome Project. I spent some time working with Harris and Jean, and I can't believe I'm forgetting people's names. It'll come to me soon. And the idea was to sequence all the known eukaryotes. There's about 1.5 million known eukaryotic species. The NIH is currently doing a program called All of Us to sequence 1 million Americans. So this size project is happening in humans right now today. And the average eukaryotic species is probably more like 2 gigabases and not 3 like a human. Currently, they're trying to get funding to sequence every eukaryotic family, a representative of them, so around 9,000 in phase 1, and they're making some progress. But I want you to imagine what you might discover from this. So one thing is our understanding of single-cell eukaryotes is actually quite poor. So there are 8 main kinds of eukaryotics, and then we're in this little group here, but we might find new ways of how eukaryotic cells work. So the eukaryotic tree is 1, 2, 3, 4, 5, 6, 7. Is still something that we don't understand, right? We don't really understand the origins of eukaryotes as well as we would like. So we would like to understand those better. There's huge things we can do with all this sequencing, right? I've talked about infectious and inherited diseases. Feeding the world is going to be a big one. New biomaterials, especially when you can take bits of different silks and perhaps join them together with AlphaFold and now simulate what a protein is going to look like. I'm at an ag school at UC Davis, right? Tamu's an ag school. You'll know that there aren't a lot of plant species feeding us. And there aren't a lot of livestock species feeding us, right? So maybe we could try and expand and protect and get ready for some amount of climate change, the species we live on, with breeding and better understanding the genetic reservoir of the non-crop species, if we can use that. The Darwin Tree of Life's been going for a while now. I haven't checked to see how they're doing, but my guess is that they've finished phase 1. They've probably done at least 3,000, 4,000 new genome species. There are only 70,000 species on the UK island. The issue is, why didn't they want to try and do more, is simply because of the politics of going to someone else's country and saying, hey, we want to sequence everything. It's not good. It sort of smacks of neocolonialism. And so they felt it was best to just start in their own backyard, and they're doing great. There is no such project like this for the USA, as far as I can tell. We tried to get one moving, but the Trump administration had, let's just say, other things on their mind. And now I think it's beginning to move again. The idea is getting through Congress. One thing I'm going to remind you of is 2/3 of eukaryotic species on the planet, if not more, are arthropods. And a lot of them are beetles and flies and butterflies and wasps and things like that. And a lot of them are really tiny too. But so I'm not going to talk about that too much. And by a lot of them are really tiny, I mean, I don't know. I'm really, this is a parasitic wasp laying on a different egg. I mean, my terrible example is the eye mite. There's mites that go in the human eye, and so they're so tiny you don't notice them on your eyelashes. And so there are some really tiny arthropods out there. But there are 3,000 of the 9,000 families are arthropods. There's no money for it yet to do the whole planet, but the costs are coming down so fast. I think it's going to take off, and it's beginning to take off. I like this quote because I went to Rice University where he said it. one annoying thing is I hear people like Elon Musk and NASA talk about sequencing and looking for life on Mars or the moon. All the good life is on this bit here. All right. So I'm hoping I told you some things. I told you the concepts are very easy. It's just the central dogma plus a lot of details. I gave you examples of human population genetics and how it might affect you. I told you that miniaturization and parallelization is what brought the cost down, and that science can be automated, right? We're generating ones and zeros and text, so we can miniaturize and parallelize. It's still difficult to predict traits from sequence, especially polygenic traits that aren't Mendelian. But now the sample numbers, and especially all of us projects, are getting to the point where we can saturate analyses. So maybe this will change moving forward. And I've told you that genes and protein sequences, especially, are the unifying data of biology, right? You can tell a fruit fly person working on para, the sodium channel, can talk to a mouse person talking on the sodium channel, and they can both talk about exactly the same orthologous gene, even if the sequences are different. And I showed you that I can tell you which genes are in insects with wings and not with insects without wings, and when that change happened. And that happened 400 or 500 million years ago. And then finally, if you ever get a chance to support the Earth Biogenome Project, please do. Maybe we should try and get all of the grasshoppers to them somehow. That's it. Far too long, sorry.
You're on time because we started 2 minutes late. So pretty much on time. So it is time for questions or any discussion. It's not like a very big group, so you can just switch on your video and mic and talk, ask the question from Fringy. Anyone?
I'll go ahead and I'll start. I'll throw you a softball question to get you started.
Okay.
But first, that was great talk. I learned a tremendous amount. And I'll go on record now to say if I don't kill the brain cells off, I plan to steal as much as I can from some of the cool ideas and concepts in the way that you presented that. So thanks. So following on from one of your cool concepts, the analogy of the car factory for producing the Drosophila knockouts was great. And I was having a discussion the other day with one of the graduate students in our lab about designing a quick and dirty RNAi experiment. And we wanted an unambiguous gene to go after. And we started talking about lethals. And so in the knockout example, there's got to be some genes in the when they abducted individuals on their way into the car factory where just nothing came out of the factory, right? Either the guy was responsible for unlocking the door or turning the power on. So do you know what proportion of roughly, just ballpark, of those 15,000 Drosophila genes were lethal?
Oh, that's a good question. There's been a lot of lethal screens done in Drosophila. It's much easier to do them on X chromosomes than the others, just the way and I do not know the answer, but I would say definitely hundreds. I don't know if it passes 1,000. I'm going to say a few hundred, but I'd actually have to look that up. I used to know shit like that. I know for and then there's just as many steriles. And there's all kinds of ways to be sterile, to be male sterile, female sterile. If I was gonna pick a gene, so you want to do an RNA knockout and you want to kill it, or
that was the idea, yeah. Something that, yeah, and especially with, like you said, protein turnover in 24 hours. If we could have a rapid lethal effect by knocking out a particular gene, that'd be great.
so I'm gonna one piece of advice I'm gonna give you, which I'm gonna copy from Monsanto, right? So Monsanto, when they started doing RNAi knockdown to try and kill things as a pesticide, and they went after, what was the first thing they went after? It was a beetle? Anyway, they said, we're not smart enough to know the answer to this question, and they tried 100 or so and picked the one that killed it fastest. Because they didn't know what would be the most effective to get into this. What the fuck was it? I forgot the species.
Rootworm, perhaps?
Yeah, something like that.
Probably was. It's a cucumber beetle.
Yeah. And so they picked 100 and they didn't know which one would get into the system best. Now, you don't have the delivery problems, or maybe you do, but you're going to use a needle, right?
Yeah.
So I would say whatever you do, look at their work, right? And don't be afraid to try 12 or 20.
All right, cool. Thanks.
Because remember, a lot of these embryonic lethals, they're early on in development, and you're trying to kill something that's already an adult.
Exactly. Yeah. Yep.
And I don't know, maybe try para.
Okay. Good deal.
Okay.
Thank you.
Okay. Anybody else? Trainees?
Vivian ran away.
No, Vivian's still there.
All right.
Okay,
It's kind of
seems,
the top of the hour. Do we declare victory?
yeah. So it seems like the student don't have much questions. So thank you very much for today to speak. I learned a lot, Fringy, and it was very fascinating to hear how you present. That's the most interesting thing. It's more complex scenarios in very simple manner. That is kind of talented you have. So you, it's very nice of you. So thank you very much for your time, and all the trainees. Thank you for joining for today crash course. I will be sharing the recording soon and also uploading to the BPRI YouTube channel. So thanks for your time. Hope to see you soon in next month if we able to get the speakers on board.
Okay. I look forward to seeing Herman tell me how to knock out all of these genes.
Okay, thanks. Thank you very much. Bye-bye.
Bye. Take care.
Take care.